All right, our next speaker is Yash Shukla. He recently completed his PhD at UT Austin, and I had the pleasure of hearing a talk from Yash at one of our he gave to our group in a group meeting on a Wednesday, and I was really, really excited by the work he was doing measuring translation in development. Such a kind of important problem for methods that are kind of studying protein synthesis outside of transcription, especially because its transcription is not so active at this point. And so I'm excited for him to tell you all about this technology because I think there's just so much fruitful crossover for people doing proteomics by mass spec. Off.
Hello. Okay, great. Awesome. Thank you so much for that introduction. I will not be talking about any mass spec, but hopefully I can convince you that a C. elegans system is a great system to study proteomics by the end of this talk. So my study looks at the landscape and regulation of mRNA translation during early C. elegans embryogenesis. So all animals start from a universal beginning when I'm showing you a one cell or a zygote of a mouse, fish, and a nematode. These undergo divisions in order to become a two cell and then finally divide to become this multicellular ball that'll go on to differentiate to become the tissues and organs that you see around you today. However, what really establishes this is this very large egg which provides these maternally deposited factors that allow early embryogenesis to happen very rapidly. So what I am studying is how maternal mRNAs regulate this early embryogenesis before zygotic genome activation. So a lot of these maternal mRNAs are present early on, and they're carefully regulated to produce protein at the right place and right time. And this happens before this maternal to zygotic switch, where the zygotic genome is activated and produces its own transcripts and proteins and so on and so forth. Now, what we know in many systems of early embryogenesis is that the mRNA amount doesn't quite correlate to the protein abundance. And for a few transcripts, we know that they're translationally regulated to control how much protein is made. But we do not know if this is a general rule or to what extent does this happen. So the question that I'm addressing is to what extent is maternally deposited mRNAs translationally regulated during early embryogenesis? And the model system that we like to use is C. elegans, primarily because it produces an invariant lineage starting from the very first cell division, where you can see in blue that there is an ectoderm that's precursor that is produced from the first division, then the mesenteroderm. And by the 8-cell stage, you have all three germlines that have been predefined. And the key thing is that these are reliant on maternal mRNA, and transcription is not involved in order for this process to occur. So now you can directly correlate translational regulation to changes that are happening in cell fate as the embryo develops. And the way we go about addressing this is by looking at this metric translational efficiency. And the way we define it is the ribosome occupancy on a given number of RNA abundance. So something that has high number of ribosomes on a given number of mRNA would have high translational efficiency, and something that has fewer ones would be low translational efficiency. So in order to do this, we had to measure two things, ribosome occupancy and RNA abundance, and that is what we did. So we took embryos at each of these stages, 1, 2, 4, and 8. And to get ribosome footprinting, we obtain these ribosomes intact on the RNA. We apply nucleases to remove all the exposed fragments and get this ribosome protected footprint. And then you can send this 30 nucleotide fragment for sequencing, and that's how you get ribosome occupancy. For total mRNA, you just collect it, and then you send it for sequencing, and you get RNA abundance. Now, for this part, single cell or single embryo RNA-seq has been done before in C. elegans embryos, and it has already been established. However, conventional ribosome profiling would require you to collect at least 10,000 embryos at each of these stages. But in C. elegans, this process happens within a matter of minutes, where they divide in 5 minutes and produce different cells. So it's really hard to actually collect those number of embryos at each of these stages. To get around this, we use this platform called RiboITP, which was previously developed on mouse embryos. This is known as ribosome profiling via isotachophoresis. And so what we do is just collect 9 embryos and then produce these fragments, ribosome protective fragments, and apply it to this microfluidics chip. and applied this to this microfluidics chip that, based off of the ionic mobility, can separate that 30 nucleotide fragment only from a few select embryos, and then you can just go ahead with library prep as usual. And so through this, we were able to move from using 10,000 embryos to all the way to 9, and that's how we got ribosome profiling data set along with RNA-seq. So we did a bunch of QC, and then finally what we want to know is, what does this data actually tell us? So we wanted to know, how does translational efficiency change as the embryo develops from 1 to 2, 2 to 4, and so on and so forth. And so here's the data. To break it down, this is the x-axis is showing you the average count or the abundance of the RNA. The y-axis is showing you the fold change, 2 cell by 1 cell. And in blue, I'm just showing you all the different RNAs, and only the significant ones have been indicated as a point in orange. And as you can see, the RNA abundance doesn't change as much from the 1 to 2 cell stage. However, if you were to look at the translational efficiency, we see these huge changes that are occurring both in up and down regulation as the embryo develops from 1 to 2. A similar pattern was seen from the 2 to 4 cell stage, where we see that there's few changes in mRNA abundance that does occur, but we see these big changes that are occurring in translational efficiency. So this tells us that overall the mRNA content is remaining constant, but it's really the translation that is being regulated of these mRNA species. So the next thing we want to understand is what drives these translational regulation. And I'm going to show you two things. First is the role of RNA binding proteins, and the second is phase separation. And those are the two things in the field that we understand could regulate translational efficiency. So in C. elegans, we have this protein called OMA1 that was previously known to regulate a few transcripts during early embryogenesis, but we did not know the extent to which it regulates it. So from what we understand is that OMA1 would bind to the 3-UTR and prevent ribosomes from binding to it. And as the embryo develops, this protein starts to degrade, and we would then observe translation of these transcripts that were regulated by OMA1. But we wanted to see the extent to which OMA1 actually regulates different transcripts. And the way we went about doing it is that we had this ribo-ITP and RNA-seq data at the wild type 1, 2, and 4. We then applied the same platform on a gain-of-function mutant in which OMA1 does not degrade over time. And then you can compare these at each of these stages to know what role does OMA1 specifically play in terms of translational regulation. What we observed was at the 1 cell stage, we don't see any difference between the wild type and the mutant in terms of translational efficiency, which makes sense because in both cases, OMA1 is not degraded. However, at the 2 cell stage, we see this large decrease in translational efficiency in the case of the mutant. But what's interesting is that in green, I'm showing you previously identified mRNA species from an OMA1 pull-down experiment. But we are observing other mRNAs that were not previously identified in those particular experiments, along with the fact that there's some upregulation that is also happening. So this specific data set of how embryogenesis is being controlled, specifically at the 2 cell stage by this RNA binding protein, was addressed through this particular experiment. And at the 4 cell stage, we see a bigger dip in translational efficiency also. Another thing that we want to look at was these phase-separated particles called P granules. So what is thought of in the field is that a lot of these mRNAs would be localized into these granules that I'm showing in green in the germline lineage, where they would trap these, what we thought was they would trap these mRNAs and prevent them from being translated. What I wanted to assess whether that was the case in our data set. And these granules are not something that's unique to C. elegans. You also see this in Drosophila or even in Xenopus. In order to address this question, we used a prior study in which people had stained 441 transcripts that localized to P granules at various different stages of embryogenesis. And the specific question we wanted to ask was to look at the subset of mRNAs that are localized uniformly at the 2 cell stage and then appear in P granules at the 4 cell stage, whether we see a dip in translational efficiency. So if localization into P granules caused this dip, causes a dip in translational efficiency, we know it's probably because it goes into these phase-separated particles. And here's something that we found that was interesting, was that the dip actually happens 1 stage before, where the translational efficiency goes down from the 1 to 2 cell stage and remains repressed at the 2, 4, and 8 cell stage. So what this tells us is that first the repression happens, and then they localize into these P granules, rather than going into P granules and causing that repression. And with that, I would like to summarize that what we've identified is translational efficiency remodeling as the embryo develops, We identified different transcripts that are regulated by OMA1. And also this repression in TE seems to be occurring prior to this phase separation. And with that, I'd like to thank my PI and my lab. And also this paper should be out in bioRxiv. So if you're interested and there's more data out there that you'd like to see, you can easily scan this. It'll be in press soon, but yeah, thank you.
Thanks so much. I'll actually start by asking a quick question. So for the sample amount requirements for doing the making the measurements, why was previously the amount, I guess, so high? Are you doing like RT-PCR of these 30 nucleotide fragments, um, or like can you not just, I guess, say you want to do single cell? Is it easy enough to kind of PCR your way to having enough fragments to sequence?
I think the reasoning is also the way you get these samples is you have to do like ultracentrifugation in order to get those particular number of ribosome footprints, and in all that process, there's so much sample loss that by the end of it, you don't have enough to get a good quantitative measurement of ribosome profiling. And so that was predominantly the reason why.
Cool. Thank you. Questions?
Very cool. I think it also reminded me of Andrew's talk with the protein dilution happening. And I was wondering if you see the similar case when you calculate translation efficiency in your ratio. Are you seeing a decrease in those transcripts that increase translation efficiency, or are you seeing a decrease in the ribosome binding?
Yeah, that would be interesting to see. I guess, unfortunately, the way we're doing this assay is we're collecting the whole embryo in itself instead of the isolated cells from there. But that's something we can do. We can isolate them and collect them and do that. But yeah, that would be interesting to see how dilution could affect the concentration.
Yes, have you been able to identify other features associated with change in ribosome occupancy, such as sequence motifs on messenger RNAs or microRNAs changing their abundance as another form of regulating translation?
Yeah, so one of the things that we also looked at was look at motifs of previously known RNA binding proteins. And I have a list of them in there, but yes, we identified, at least for the ones where the data was available for this particular motifs, of how much do they correlate with the translational efficiency that we see. We didn't yet do like a motif search to identify which motifs explain higher translational efficiency. That would be sort of a next step that we want to take.
Cool. Well, one other quick question with the RNA binding proteins that seem to affect translation. What causes that change? Because presumably that RNA binding protein was present in the zygote. Do you have thoughts as to what results in the switch?
Yeah, so one of the things that I have two reasons, three that I can think of on top of my head, and for different proteins there would be different contexts. One of the things that happens during this early embryogenesis is there's a lot of asymmetric divisions in which the RNA binding proteins are divided separately. And so depending on which RNA binding protein is present in what stoichiometry, you can define the TE of that particular transcript. There is a lot of phosphorylation and changes that are happening on these particular mRNA binding proteins that regulates the translational efficiency of these different mRNAs. So those are a few that are on the top of my head.
Conceptually possible mechanisms, but it's not necessarily clear for any one particular protein what is driving it.
That is correct. I think that people have not really dissected yet, and I hope that through this data set, people can start really like looking at RNA binding proteins and what are the different species that it regulates together. Yeah.
Thank you.
One additional quick question: Is RNA degradation like muted in some way at that 1 to kind of 2 cell stage, since you kind of need those early deposited? Which, since you showed those plots showing that like the actual mRNA abundance does not change substantially from like the 1 to the 2 cell stage, and so presumably, I guess if they were kind of being degraded at different rates, you would kind of naturally see maybe a lower, because since there's no transcription happening, a decreased abundance of mRNAs that degrade easier. So do you know if maybe this process is not happening in addition to lack of transcription?
Yeah, from all the other data sets that I've looked at have done RNA-seq, the RNA amount stays fairly constant from 1 to 4 cell stage, and it's really after that a lot of degradation starts happening in different cell types. So I guess from the resolution that I have, it seems like it stays fairly stable.
Thanks. All right. There are no more questions. Sorry. Oh, there's one online. Another localization. Wow. Thanks. Yeah, someone is asking about the localization effects other than P granules. So outside of that.
Yes, apart from P granules, we looked at transcripts that are localized on the periphery of the cell, and all of them have a higher translational efficiency relative to the average. And also in terms of localization, we've also looked at transcripts that are localized in the somatic lineage versus the germline lineage. And we observed that in the germline lineage, they seem to be repressed compared to the somatic lineage. So those are the two other localization things that we saw apart from P granules.
Well, in the periphery, do you mean like near the cell surface?
Like on the cell cortex, yes.
Do you think that there are RNA binding proteins that are like somehow also transmembrane or localized to the surface in some way, or maybe interact with things on the surface that could?
For some of the transcripts that people have previously studied, what ends up happening is they get partially translated, resulting in a domain, like a domain that binds to the membrane, and then they finish their translation on the membrane in itself. So that's where I think that active translation is happening for them.
Okay, cool.
Yeah.
Thank you. Interesting.